Well, hi everyone, I'm Diba and I'm 17 years old. So when COVID started 2 years ago, I was, I think, in grade 9, and I had a lot of free time to just do something new. And I wanted to learn about artificial intelligence and other emerging technologies and how they can be used to solve some of the world's problems. At the same time, in the news, there was a lot about a COVID treatment or vaccine being developed. And luckily, a few months after that, it came out in the news that remdesivir was being repurposed as a drug for a treatment for COVID. So in this presentation, I also wanted to talk about the use of artificial intelligence in drug repurposing and a project I worked on where I tried to find a new way of applying artificial intelligence to drug repurposing for a new purpose. So drug repurposing, briefly, it's the repositioning of an approved or failed or still being investigated drug by FDA for a new disease indication. And it's not something new. 30% of drugs and vaccines approved by the FDA today are repurposed, and repurposed drugs generate around 20% of the revenue for the pharmaceuticals industry. So the benefits of drug repurposing is it takes less time, much less money, and has a lower risk of failure as well because of the lower amount of stages it requires for development. Drug repurposing can happen in two ways, either an on-target strategy or an off-target strategy. So with on-target, the pharmacological mechanism of a drug is known. For example, minoxidil was previously an antihypertensive vasodilator, and given that the biological pathways that it affected were known, then it was repurposed as an anti-hair loss drug. Off-target strategy is when the pharmacological mechanism is not known. An example of this is aspirin, which was an anti-inflammatory drug, still is. And at the time that it was going to be repurposed, it was unknown how it could treat heart attacks and strokes, but it did. Today, we know the pharmacological mechanisms. So about machine learning is a subtype of artificial intelligence. Artificial intelligence is quite a vague term because the wide variety of automated tasks that exist now, but machine learning is one subtype where a machine learns to carry out a task on its own. So the benefits of machine learning is it's much more efficient and quicker than humans. For example, if you're looking for a ring that's lost in sand, it's much easier to use a metal detector than just like your bare eyes. So that's the same with machine learning and trying to find like a drug compound that works well for a disease. It's much easier to use machine learning. You can teach a machine to carry out a task in three ways: supervised, unsupervised, and reinforcement learning. With supervised learning, the one that's until now, it has been the most commonly used type of machine learning in drug discovery. There is labeled data that's given to the machine. So take the example of fruits. You have pictures of oranges, pears, and apples, and the name of those fruits are also given to the machine. So when it's given the new data that it hasn't seen before, it can predict the name of that fruit. With unsupervised learning, the label is not given, just the images. And so the model will learn to pick up on patterns and similarities between images and then group them together. So when it sees a new image, it can then put it into similar groups. It doesn't have to be an image all the time. This is just an example of the fruits. With reinforcement learning, the model will carry out a certain task, and if that task is correct, then it's going to build on top of that. But if that task is not correct, then it's going to go back to state 0 and redo that task, sort of like a child learning to behave well. Going deeper into supervised learning, there's two types: regression and classification. With regression, this is simple regression where the model will learn to fit the data into a regression model and then make a numerical prediction from that. With classification, a numerical prediction is not the final output. Rather, it's the name of the class or a group that the data belongs to. For example, in the case of the fruits, then it's going to be either pear or apple or oranges, but not a number. In drug repurposing, bioinformatics and cheminformatics are commonly used. Usually there's virtual high throughput screening, which is used It can be done through structure-based or ligand-based. With structure-based, when the 3D structure of a target protein is known, then molecular docking can be done. With ligand-based, when the 3D structure of the protein target protein is not known, then a Qsar modeling can be used. Another way bioinformatics can be used in drug repurposing is gene alignment. So that's when the target protein that a compound targets is going to be studied on a genomics level to find similar genes to that. And the purpose of that is to find similar proteins to that target protein that a drug can then be also used for. Going deeper into Qsar modeling or quantitative structure activity relationship modeling. This is a computational model that seeks to find a relationship between the structure of a compound and its biological activity. So for example, the biological activity can be defined as IC50 value or any other value that can define some sort of biological activity here. And you have a large database of molecules with their structure and their biological activity value. And then the model will try to narrow it down to the compound with the desired biological activity. But there's limitations to Qsar modeling too. When you want to have that model learn the structure of the compound, there's just so many descriptors to use. So one problem with Qsar modeling is it only gets a narrow view of the molecular structure of a compound. Second problem is for the computer to understand molecular structure, you can't just feed it like a Bohr-Rutherford model or something that is comprehensible by humans. A binary system of fingerprints can be used, which consists of zeros and ones. But because of that binary system, a lot of detail about the structure is lost to those zeros and ones. And then also correlation is found between features where there is no correlation just because of the narrow view that the model gets of the molecular structure of a compound. Qsar modeling can be coupled with machine learning to overcome some of those limitations. So some common ways that machine learning can be applied is linear regression, which is simple linear fit, and then Bayesian neural networks, which use Bayesian inferences in addition to neural networks to quantify uncertainties. And lastly, random forest, which uses a bunch of decision trees in order to make a majority vote at the end so that the result is more reliable. For example, it's like imagining a bunch of legislators who agree upon one legislation together. So there is much more reliability in that final decision instead of just one person making that legislation. The problem that interested me when it came to drug repurposing was the lack of access and availability of antiretroviral drugs in African countries for those with HIV. So to this day, 36% of HIV patients in African countries are missing access to ART drugs. And during the COVID pandemic, availability was reduced even more, especially in South Africa. Actually, quite a few years ago, an interesting initiative was taken to reduce the cost of HIV treatment for patients in African countries. And so the cost of ART drugs was reduced to around $70 to $80 per patient for an annual supply. And this is wonderful. I mean, in North America, that is a very like $70 to $80 is so much smaller than what patients in North America have to pay. But the thing is, these ART drugs are produced in India and then imported to African countries. In 2015, around 98% of ART was imported to Africa. Some countries like Zimbabwe and Uganda have started local production, but there's still a heavy reliance on importation. And with importation, there's problems of bottlenecks of how much product can be imported. And also when there's a crisis like the COVID pandemic, where supply chain is completely messed up. So I asked myself, why these African countries are not locally producing their own ART drugs, because a lot of other countries are doing that. And the thing is that for ART drugs, their active pharmaceutical ingredients cost a lot more than other types of drugs. So for ART, the API costs around $120 to $800 per kilogram. Whereas with glyburide, a drug for diabetes, it costs around $43.1 to $115.2 per kilogram. So a hypothesis I had was, what if we repurpose drugs that have a lower, that have a cheaper API cost to be able to treat HIV, so that countries that do not have the resources to produce the current ART drugs can then locally produce it and avoid having that reliance on importation and the limits that importation poses on it. So I wanted to do this using machine learning, and I used resources that were available to me. So the model I wanted to use was LightGBM because it was optimized for accuracy and speed. LightGBM is based on a decision tree that grows the leaves with the maximum delta loss. So at delta loss, I mean the most inaccurate prediction that the model makes. So the model will try to make the most inaccurate prediction as accurate as possible. So LightGBM is very much optimized for accuracy. And one problem it has is it is prone to overfitting when there's a smaller data set. It's also a fast model. It runs pretty fast because it doesn't require that much memory. So it's great when there is low computational resources available. So I then coupled this LightGBM model with Qsar modeling principles, where I tried to find a relationship between the molecular structure and the biological activity. So the molecular structure, I defined it as the SMILES strings, and the biological activity that I chose was IC50 values, which is a way to quantify potency of a drug. More specifically, I took the PIC50 value, which is the negative log of IC50, just for better comprehensibility by the computer. And then the database of compounds I used was from CHEML database, which is commonly used in cheminformatics. The target proteins that I wanted to work with were, first of all, the HIV reverse transcriptase, an enzyme which is crucial for the virus to continue infecting a host. And then the second target was the CC chemokine receptor type 5, which is actually a receptor on human white blood cells that allows the HIV virus to enter human white blood cells and infect it. Current ART drugs are mostly for HIV reverse transcriptase and protease, so elements on the virus itself, but a new class of ART drugs are being developed that target CCR5. So I wanted to work with targets for old and new types of ART drugs. The workflow I used was, first of all, I pre-processed training data. So I had the molecular structure, which I then defined as SMILES, and fingerprints that were understandable by the computer, and then the IC50 values defined as PIC50. And then I made a very small data set of test data, around 4 compounds only, just because this model I was working with was a very simple prototype, and I just wanted to test if such sort of a solution would even be possible for drug repurposing. And for the test data, I only had the molecular structure and not the biological activity. Then I used a library called LazyPredict to test out a bunch of regressors to find the one with the highest accuracy and lowest speed for training. And here I found that LightGBM was the best regressor for me to use based on the criteria I set for myself. And I trained the regressor and I tested my test data on it. The framework I used was developed by Chan and Nantasamat. So here this slide shows the accuracy of the model. So because there was a large number of features in the model, I wasn't able to quantify the accuracy, but I visually represented here where the predicted PIC50 value is compared to the experimental PIC50. And they're in a close range, but the R-square value is around 0.46, which is not extremely high. But the MSLE, the error was 0.03, so it is on the lower end a bit. And when I tested the test data on it, I set up some cutoffs. So a PIC50 value that was higher than 6 was considered as active, and a PIC50 value under 5 was considered as inactive. And so I found that Glyburide was the only cheap API drug active for reverse transcriptase. Aspirin was inactive, and metformin and ibuprofen were intermediate. Now for CCR5, the predicted PIC50 values and experimental PIC50 values were in a much closer range than for reverse transcriptase. So the accuracy was a bit higher here. The R-square value was also higher, around 0.67, and MSLE was also lower, 0.01. Once more, Glyburide was the only cheap API drug active for CCR5. Aspirin, metformin, and ibuprofen were intermediate. So what is Glyburide? It is a drug that's used to treat diabetes mellitus type 2. It is a potassium ATP channel blocker and broad spectrum ATP binding acid transporter inhibitor. I wanted to dig deeper into literature to see if anyone has found any relation between Glyburide and HIV, and there isn't much out there. There was one study I found by Code 2011 where it was indicated that Glyburide inhibits inflammasome assembly, and also in their study they found that patients with human melioidosis who were on Glyburide had reduced mortality, and so they correlated this with an anti-inflammatory effect of Glyburide. In another study by Monroe in 2015, where they were looking for special considerations for HIV patients with diabetes comorbidities who were taking diabetes drugs also, and they found no special considerations for Glyburide alongside ART drugs used for HIV. Then I also wanted to do a quick molecular docking analysis. I used the MCULE docking platform, and the most negative value I found for reverse transcriptase and Glyburide was -5.6. Least negative was -4.5, which is not extremely negative, but it seems to be in an adequate range. But to make this project go beyond a prototype, because this was a very simple workflow I used just to test the hypothesis of, will it be possible for us to repurpose drugs with the goal of lowering the API costs to promote local production of certain drugs in under-resourced areas. And some of the ways that this can go beyond prototype is by using more than one regression model and using more than one biological activity indicator. So using either like a random forest model or a Bayesian neural network alongside any other value other than IC50. Also, for the test data, instead of using only 4 experimental drugs, it's preferable if there's at least 100 drugs being tested. And then the model I used was optimized for accuracy over reliability. So one way to also optimize for reliability is to use a random forest model, again, because of how I explained that there is multiple decision trees working together. And if a light GBM model is being used, because it is prone to overfitting, I had around 7,000 rows of data for reverse transcriptase and around 2,600 for CCR5. One of the ways to avoid overfitting is to do deeper hyperparameter optimization. Other than that, a deeper molecular docking analysis would also give more insights into the structure of reverse transcriptase, CCR5, when the Glyburide comes in contact, and also integrating this with in vitro experiments, because everything I did was just in silico. So thank you for joining today. I'm Personally, I really find the topic of drug repurposing interesting because of the possibilities when it comes to undiscovered disease indications. And if you want to follow my projects with drug repurposing and artificial intelligence, I'm available on LinkedIn, YouTube, and Medium at Diba Dindoust. And I'd be glad to answer any questions now or chat later.
Thanks, Deva, for your presentation. Now, floor is open for the questions.
Yep.
Dr. Bruce, I can.
Hi, Diba. Well, I'm just blown away by what you've done. Your knowledge about AI and machine learning is way better than mine. So I'm curious where you figured out, how did you learn all of this stuff? I mean, you're in high school. Is there like a class on AI machine learning, or do you have some mentors? Tell me a little bit about that. I'm curious.
Yeah, so I'm in grade 11 right now, and I'm not a computer science student at all. It's not something that really interested me until recently, when I joined this program called the Knowledge Society. So it's an after-school program where students from 13 to 17 are exposed to emerging technologies, but it's very much an independent process of learning about AI and gene editing, blockchain, all these other really cool technologies that are out there right now. And for me, I worked with a pharmaceuticals company over the summer last year, and I really saw how interesting it would be to go deeper into the computational side of things. So in September, I tried to learn everything on my own. So resources I used were articles on Medium and YouTube. And obviously, it's really great when people in the field answer cold outreach calls and are willing to share their advice and their expertise. So I really have some great mentors to help with that as well.
I will connect with you on LinkedIn. There may be some other people I can connect you to.
Thank you. That'd be wonderful.
Thanks, Diva.